Welcome to part 2 of my lesson on cell structure. In this lesson, we're going to look at specialised cells and stem cells. Animals and plants produced by sexual reproduction begin life as a single cell, a fertilised egg or zygote. This cell must divide to produce a multicellular organism. It must also differentiate so that cells develop features that enable them to fulfil specific roles. You need to know a variety of these specialised cells for your course, so we'll start with the red blood cell, which is found in the blood. Its role is to carry oxygen around the body. It has no nucleus to give more room for haemoglobin, which binds with oxygen to carry oxygen around the body. It also has a biconcave shape, which gives it a large surface area for absorbing oxygen. Next are the ciliated epithelial cells. These have little hairs on them, and their role is to waft things along. They're found in the oviduct, where they waft the egg along, and also in the trachea. In the trachea, they work with goblet cells, which produce mucus to trap dust and microbes. Their hairs on the ciliated epithelial cells waft that mucus containing the dust and the bacteria up to the back of your throat, where you then swallow it into your stomach, where the bacteria are killed. Next are the egg cells, or the ova. These are produced in the ovary, and their role is to get fertilised by sperm. They have a jelly layer on the outside which hardens after the first sperm has got through, and this prevents any further sperm getting through. They also have a haploid nucleus, which means it contains half the normal number of chromosomes. This means that when they are fertilised by the sperm, that the normal number of chromosomes are restored. After the egg comes the sperm. The sperm are produced in the testes, and their role is to fertilise the egg. They have lots of different adaptations. First of all, they have an acrosome, which contains digestive enzymes to digest through the membrane of the egg. They also have a haploid nucleus, containing half the normal number of chromosomes, so that when they fertilise the egg, then they restore the normal number of chromosomes. They also have lots of mitochondria in the midpiece, which provides energy for swimming, and they have a long tail for swimming to the egg. Now we move on to the plant cells. Palisade cells are found at the top of the leaf nearest the sun, and their role is to photosynthesize. They are columnar in shape, so packed closely together, and they are packed with chloroplasts, which are the site of photosynthesis, so that they can produce as much food as possible. Root hair cells are found in the plant root, and their role is to absorb water and minerals. They have a long, thin extension to increase their surface area for absorption. Guard cells are found mainly in the bottom surface of the leaf, and their role is to open and close the stomata, which are tiny holes in the leaf. Their adaptation is that the inner cell wall is thicker than the outer cell wall, so when they swell with water, they bend outwards like a sausage, opening the stoma, which is a singular version of stomata, in the middle of the guard cells. Cells which have not differentiated yet and become specialised cells are known as stem cells. A stem cell has the ability to divide many times while remaining undifferentiated. And there are two types: embryonic stem cells, which can differentiate into any type of cell and are known as totipotent, or adult stem cells, which differentiate into several types of cells, known as pluripotent, and examples of these are bone marrow and skin stem cells. There is much potential of using stem cells in medicine, especially embryonic stem cells that can differentiate into any type of cell. The use of stem cells in this way is known as stem cell therapy, and the most common use so far is for bone marrow transplants. Bone marrow and other adult stem cells are readily available, but they have limited ability because they can only differentiate it into certain types of cells. The best way to use stem cells would be to use embryonic stem cells, which can differentiate into any type of cell. There is a lot of research going into this, and there is hope that they can be used to treat diabetes type 1, Parkinson's disease, heart disease, or even paralysis. However, we have a long way to go, and there are many people who object morally to using these cells for medical purposes. Stem cells are also found in plants. Here they are found in regions called meristems. The stem cells of plants do not lose their ability to differentiate into any type of plant cell, unlike in adult animals. Meristems are mainly found in the tip of the shoot and root. As the cells mature, they enlarge, differentiate, and develop a large permanent vacuole. Now let's look at the advantages and disadvantages of using stem cells. They could be used to grow organs that could be used in transplants. They could be used to develop treatments for diseases such as Parkinson's disease, Alzheimer's, cancer and diabetes. They could be used to study the stages of development of the human embryo, so work on prevention of birth defects, pregnancy loss and infertility. They could be used to test new drugs directly on human cells without the need of animal testing. And the use of adult stem cells to treat disease means that the patient's own cells could be used, which reduces the risk of rejection. However, there are disadvantages. It involves the destruction of human embryos, and for those people who believe that life begins at conception, to do this is immoral and unacceptable. Like any other new technology, the long-term effects are unknown. There has been some evidence of heart patients treated with stem cells ending up with narrowing of the coronary arteries, for example. The use of embryonic stem cells means the cells are not usually the patient's own, so there is still a risk of rejection. And the use of adult stem cells is restricted due to them only developing into a limited number of specialised cells. We have now completed the section 2.2 on cell structure. We have looked at the cell structures and their functions, including the nucleus, cytoplasm, cell membrane, cell wall, mitochondria, chloroplasts, ribosomes and vacuole. We've compared plant and animal cells. We've looked at cell differentiation and some examples of specialised cells. We've also explored the advantages and disadvantages of using stem cells in medicine. Hopefully you found this lesson useful. Please do leave any feedback you think would be helpful in the development of my videos, and please do subscribe to my channel.